So in terms of the exam, are there questions about any of the material? But we do have some time, so this would be a good time if people have questions, if they want any clarifications about the material.
I don't have questions necessarily about the material, but how is our exam structured?
It's going to be maybe like it's going to be in Proctorio, online. It'll be a mix of kind of short essay questions. Essay is pushing it, like short answer questions, where, kind of like what I asked on the warm up today, like what are different ways you have for clearing a synapse? Or it could be questions like, there could also be, multiple choice things like things I was asking about graded in action potentials. Or I could have put that as a short answer, like, compare and contrast graded versus action potentials and lay that out for me, but use your words. So it'll be a mix of both of those. It'll be a mix of kind of multiple choice kind of questions, as well as these short answer kinds of questions. And it'll be designed to, last, like for, 90 minutes. So, what else? Yeah, and that's how I would describe it.
Are we going to be logged into Zoom while we take it, or do you just want us to start the exam at 8:10 like we would log into Zoom?
No, you should come in. We're going to start the class. You should log into Zoom.
Okay.
Log into Zoom before we get started, and we'll talk about, because we actually are going to talk about we are going to have a lab after the exam is done. So the lab is part of the day, but then, it doesn't take up 4.5 hours. So.
Are you basically giving us the entire time of the lecture for the exam?
Okay. Yeah, exactly. And then what else? Yeah, are there other questions about the exam?
I have a question.
Mm-hmm.
It's actually a question that we had on a warm-up, and I am some foggy on some lectures, so I'm going to go over them. But this question I'm confused about. It says, if a cell had a membrane voltage of -70 membrane voltage, if we are just considering the force due to the electrical potential, a cation such as sodium will tend to, and the answer is move into the cell. Why would it move into the cell?
Okay, so this is important to. So let's look at our cell. So the main thing to remember is when we talk about the membrane voltage, it's always going to be the inside of the cell compared to the outside. So voltage is always inside cell with respect to outside. That's just the convention. So if I have a cell that has a negative voltage, if I say V sub M equals -90 millivolts, that means that this -90 means the inside is more negative compared to the outside. That's why it gets a -90 millivolts. If it was the same amount of difference, but positive inside compared to the outside, it would have been a +90 millivolts. But because it's -90 millivolts, it means the charge separation is such that there is more negative inside compared to outside. Again, it's always the inside with respect to the outside. So So then we just think about a cation. And we think about if we're just going to be imagining electrical, electrical things, charges are attracted to opposite charges, right? Opposites attract. So which way does this cation want to go?
Inside.
It wants to go in to where it's more negative.
Okay, so then if it was a positive 90, it'd be more attracted.
Then it would want to go outside because it would be more negative inside and it would be positive inside. So it would be kind of repelled from there.
Okay, okay, now thank you.
Sure. So, right, so that, I mean, that, and again, the warm-ups, part of the reason I make sure that the warm-ups stay accessible to you. You know, that question could easily be on the exam. Anything that's in a warm-up is something that could be on an exam question. And so much more. Yeah, so that's a good question. Any other questions?
How many questions do you know?
I'm not sure. Yeah, it's 100 points worth, but
Okay, thank you.
yeah, I don't know.
Can we just go over the primary active transport and the secondary active transport again?
Uh-huh. So in both cases, active transport is going to mean you're using energy of some sort. And you're using the energy to move things against their concentration gradient. But where the energy comes from to do that is different. So primary active transport is the simplest one. This just uses ATP to power things. So for instance, if I am pushing sodium out, even though there's already a lot of sodium outside, and pushing potassium in, even though there's already potassium in, it doesn't want to go that way due to its diffusion. We need energy to make it happen. So in the case of this little pump here, we are just taking ATP, breaking it apart into ADP. And that energy is used to run that pump. So this is primary active. It's just some little pump that burns ATP directly in order to drive the motion. So secondary active. This one's kind of more complicated. So the example I gave was we wanted to pull glucose into the cell against its concentration gradient. So we have this thing here that lets glucose in, but it's not gonna, and it needs energy to pull the glucose in to make this happen. So, but where that energy comes from is from a gradient of some other molecule. So we have like this symport here. So here my sodium gradient, sodium concentration gradient, provides energy to move the glucose inside into the cell. You know, that means, you know, the fact that we have a sodium gradient means we must have been making it happen. So in the meantime, we also have some primary active transport going on over here, where we're pumping sodium out when we're using ATP. Wait, I should yellow. Whoops, sorry about that. So over here, this is primary active transport. Sets up the sodium gradient. But then over here, this is going to be secondary active transport, which is being powered by the sodium gradient that exists.
Thank you.
What is ADP and the PI? What does that stand for?
Uh, so ATP, what does ATP stand for?
Adenosine triphosphate?
Mhm. And it's an A with 3 phosphates. So when it breaks apart, it becomes, so this is ADP, this is di. Di, which just means two. Like dichotomy is two sides of something. Dihedral is between the two wings, or diptych, lots of things, right? Depending whether it's Latin or Greek, it can be di or bi. These are things that mean two, right? Sometimes it's like a bicycle, but sometimes it's a diphosphate. And then this P is just as when you have a phosphate that's not connected to a carbon containing molecule, then it's an inorganic phosphate. It's just the convention. When you write the breakdown of ATP, you do its ADP, which is self-explanatory. It's just A plus the 2 P's. And then the lonesome P on its own, that's no longer connected to an organic part. And now that's why it's got the subscript of I, because it's now just a phosphate group no longer connected to an organic. That's why it's an inorganic phosphate. So it's just the conventions that people use, and obviously there's always going to be energy also when we hydrolyze the ATP.
I remember seeing that equation in the electron transport chain system when I was going through the synthase, I think. What was the point of using ATP in that part of the reaction?
The synthase, that's actually the opposite of that. That's running this backwards, right? That's how we make ATP. That's using the energy stored in the proton gradient to take ADP plus phosphate and make ATP, which is the final goal of cellular respiration. So these are the building blocks that you assemble to make the ATP if you're running it backwards. Right, most of the time you're actually burning ATP to power cellular processes, but obviously in the mitochondria you are making ATP using cellular respiration.
I have a question to piggyback off of that. Once the cell has made enough ATP, does it immediately start trying to store the energy as fat, or does it use other like lower energy models or molecules? Sorry.
It depends, but yeah, a lot of times you turn on pathways that just start, assembling fat molecules. That's like a very common thing to do. If you've got enough ATP, you can just, instead of taking acetyl-CoA and putting it into Krebs cycle, you can just start assembling these hydrocarbon chains and make fat for storing energy for a rainy day.
Okay, because I know the cell obviously wants to keep that gradient favorable, so it'll keep pumping out ADP or ATP. But
right? But if you like, as you've probably seen, like if somebody just keeps eating lots of high energy food, at some point the body has more than enough ATP and it starts just, people start, storing it away as fat on their body.
Okay, that makes sense.
Right, there's a minute there, you need to have enough ATP, but particularly in kind of our modern Western, I mean, it's kind of even in the across, yeah, even in like within our community, there are people who have, problems with food, but in general, Americans are pretty imbalanced about taking in more food than they need. There's a lot of obesity in the United States. Um,
I have two questions. One, it's pretty self-explanatory. Is there going to be a bigger portion of the chemistry review on this exam? And then my second question was if you could just kind of explain again the excitatory and inhibitory postsynaptic potential.
uh-huh. So, I would say the chemistry stuff is going to be proportional to like how much time we spent in the class, right? We spent the first 2 weeks talking about that stuff. So, it's important, so it'll definitely be, a real part of the exam. That being said, we also spent a lot of time looking at cellular respiration and at all the, transepithelial transport and action potentials and graded potentials. So that's all going to be important too. I would say go to that Appendix A review thing, and it will kind of give you a sense of all the different things that would be fair game, as well as looking at your notes. And then the other question. Oh, wait. Oh, no, I just passed. Say that again.
It was the excitatory and inhibitory postsynaptic potential.
IPSPs and EPSPs. So this was talking about a synapse. So we've got, here's some neuron. There's like some dendrite on this neuron. Here's this is gonna go. So here's my neuron. Back. And then I have some other neuron. It's axon and little telodendria, and it's making a synapse here. And then if we think about what happens at the synapse, we have some neurotransmitter that is released and binds to receptors on the postsynaptic membrane. So that's what's going to happen. We have action potential comes down this dude, gets to the end here. We release neurotransmitter. It drifts across the cleft. Receptors bind the neurotransmitter. And now what happens? What happens once the receptors on our target neuron are binding the neurotransmitter?
You open the ion channels, right?
So
and whether if it is excitatory or inhibitory, if it is excitatory, it will trigger another action potential, right? Would it?
right. Well, so let's before we get there, let's just talk about, we open ion channels. It depends what kind of ion channels we open.
Correct.
If we open up like a sodium channel, that is, the membrane potential is going to go up. Sodium will come in, the voltage will go up. That's what we call excitatory postsynaptic potential. Postsynaptic is just the potential on the membrane after the synapse. So if we're letting in like cations, for instance, like sodium, we're going to have the voltage go up. We'll say it's excitatory because we are bringing this target cell up closer to threshold where it might send off its own action potential. If I open up chloride channels though, and I have anions come in, I'm going to have the membrane voltage go down. That's what I'm going to call an inhibitory postsynaptic potential. All of a sudden, this voltage on this cell is getting lower, so it's getting further away from threshold. It's less likely to have an action potential. So that's what I mean by an EPSP or an IPSP. It's like when you activate the synapse, if it's an excitatory postsynaptic potential, it's opening channels that increase the voltage and bring our neuron closer to threshold. If it's an IPSP, we are opening channels that make the membrane voltage more negative, and we are taking it further down away from threshold, making it less excitable. So does that make it clearer?
Yeah, thank you.
Okay. Yeah, and you are not, I feel kind of sad. One of the things that normally, for 20 years, we've had to give you for like study guides, all this, it's like the stuff that we can't do anymore because Adobe Shockwave has been discontinued, but they have these little quizzes, and if you get them right, the neurons do these cool little dances and stuff like that. And now I can imagine them in my mind, but you'll never see them. It's too bad.
For me, is there a way you can go again quickly over the transepithelial transport?
So that is kind of connected to
Primary and secondary active transport.
exactly. So let's take a look at that. Yeah, so transepithelial transport is really important. We're going to be seeing it as the semester goes on quite a few times. It's going to be at the core of absorbing nutrients from your digestive system. It's going to be at the core of processing in your kidney tubules. So the idea is there's an epithelial sheet of cells. So here I'm drawing these columnar cells. So
12 p.m.
So here's my epithelial cells. And the idea is there's something on one side. I did the example of absorbing glucose in the digestive system. Let's say this is the lumen of your intestine. And there's some glucose molecules. And if I just leave them in the lumen of the intestine, they're going to end up in your poop. You don't want to poop them out. You want to absorb them into your bloodstream so you can use them. So on the other side of this epithelial sheet, we have like capillaries. So the trick is I have the glucose over here, but I want it on this side. And it's not going to happen automatically. We need to use active transport and other kinds. So it's going to start with that secondary active transport, like when I was answering Grace's question. Right? So we have like that symport that couples glucose coming in to the movement of sodium coming in. Right? And this is going to be secondary active transport because it's driven by the sodium gradient. which means we also need to set up that sodium gradient, which is going to be primary active transport. Right, so just in this picture here, we've got a couple of things that are important. Over here, we've got secondary active transport. It pulls the glucose in using energy from the sodium gradient. But then we set that up using primary active transport, using the power stored in ATP to pump the sodium out of the cell. So this is going to bring the glucose into the cell, but we still need to get it out the other side to where the capillary is. And that's going to be another membrane protein, but this time just a glucose carrier. Because glucose here can just go down its concentration gradient. We're bringing in all this glucose. Any glucose on this side is just getting taken up into the bloodstream. So we have a concentration gradient where glucose wants to leave the cell. Glucose needs to have a carrier in order to actually cross the membrane, because glucose is a large polar molecule. So what do we call that when you have glucose crossing the membrane along its concentration gradient, but using a carrier?
Facilitated diffusion.
Exactly. So this would be facilitated diffusion.
But I think my thing is this. So sodium, when it's in the lumen of the intestine, wants to go into the cell because of the low concentration that is there in the cell. And so
Yeah.
it brings glucose with it as a import.
Yes.
Okay. And then active transport is used for sodium to, okay, I get it.
Yeah, so the active transport creates the sodium gradient down here. And then we use the energy of that sodium gradient to do secondary active transport up here. And then we talked about how do we make sure that stuff doesn't wiggle in between, that you would rather not, that you're only moving the stuff intentionally.
Tight junctions.
Yeah, exactly. Remember we had like those tight junctions. So if anything tries to wiggle in, it can't. So tight junctions make sure that you have a kind of tightly sealed boundary between the cell membranes. So only the stuff that's moving according to the plan is moving from one side to the other side. Yeah, I mean, I promise you there'll be questions about transepithelial transport. This is an important concept that we'll be revisiting when we get into different systems.
And so this type of transport basically uses all types, like primary, secondary, passive, facilitated.
Yeah, those are all involved in describing this overall transepithelial transport situation.
I have a question about the secondary active transport. Can that also use an antiport, or does it have to be a symport?
I mean, theoretically, I guess it could use either kind, but I think, let me say, I usually think of it this way, but that doesn't mean that there aren't other more complicated situations of it. But I don't want to say anything definitively and be wrong. So I wouldn't be surprised, I would say, but the examples that come to my mind are like this. other questions about stuff?
Yeah, one more question about this enzyme lab. Do you want us to print out that data, or are just the graphs sufficient for the data that you require?
So that's I think it's an interesting one, because normally you write your own data. I think it's probably not a bad idea to at least have the average numbers so we can see what we're working with. But yeah, no, it's in general, I don't have you print out every single number from every single group. That usually seems silly. So I would say I don't know. Because again, we're in COVID time, so this is not normally how we do this. So it's kind of weird. Yeah, I would say use your judgment and do what you think feels best. But yeah, it probably doesn't make sense to print out every single number because it's not really helping anything. Yeah, sorry, I wish I was less wishy-washy there.
Professor, I had a question. You remember for the last warm-up, I think warm-up 6, I'd asked you, there was kind of like contradictory information from the textbook about the second.
Oh, second, right. I did not look at that. Here, let me look at it.
Go off your word. I just want to be sure.
Yeah. Do you remember what page was that again?
I think I said 161.
Let me see what she writes. No, not 161. Let's see, let me look on 180 here.
Oh yeah, 181, that was it. And the figure B. That kind of confused me.
Right. So the second messenger molecules would be like cyclic AMP or cyclic GMP or calcium. That's what we'd fit in that red box there. And then those activate the protein kinases and stuff. Although then they're saying increase intracellular calcium, so that's kind of weird. Yeah, the kinases are not the second messenger, though. Those are things that are activated by the second messenger.
Okay, and is the PDE also activated by the second messenger?
Wait.
The phosphodiesterase?
The phosphodiesterase is a thing that breaks down the second messenger.
Okay, that's what I thought.
Right, it's the stuff that actually stops the pathway, turns it off.
Okay.
Right, but then, like I was saying, it can get more complicated because you can have some other second messenger pathway that's turning on PDE to influence the activity of another pathway.
Okay.
Yeah, and then, the reality is you often have one receptor turning on more than one second messenger pathway. And that's when we start talking, the reason different drugs can have different effects is like how they affect what kind of second messenger pathways get turned on and what proportions can actually. So the idea of agonists or antagonists gets actually way more complicated even because different kinds of exogenous ligands can actually do weird things with how they affect the second messenger pathways that are turned on by the receptors. But
Okay. Thank you.
all right. Questions, comments?
My notes for the advantages of second messengers is a little messy. I just want to make sure I have the right main ones. So I have increasing control, flexibility, and amplification of a signal. Is there any other ones that I'm missing?
No, although you want to make sure you know what, like if you say flexibility, what do you mean by flexibility?
That the same receptor can have different outcomes and different receptors can result in the same outcome.
Exactly. Yeah. So that is that. Basically, we have that kind of flexibility. The amplification is huge. And again, just having that ability to have a lot of control to can have finesse in what's going on and how different processes can influence other processes. Definitely, it always cracks me up. I'll ask a question on an exam about what are advantages, and they'll say like, oh, it's, what is it, like, it's less complicated. It's like, no, it's way more complicated than think about a directly gated like nicotinic receptor. It's like bind, open, you're done. It's way, it's way messier, but it does have advantages beyond being, so what else? That's usually more when people are like walking into the exam. It's like, ah! So I would say, let me give you just a few things. Relax. When you have like, it's useful to breathe. Like, cleansing breaths are good. Breathe, cleansing breaths. Having the ability we do this, actually, I do this with my subjects, like in the studies, like people are getting kind of like anxious. You just like, you breathe in and you kind of hold it and you slowly breathe out. And it can actually really kind of, your brain's spinning around and you kind of come back and just do a nice calm breath can actually help like de-escalate wherever kind of place you're spinning out into. Relax. Reread the questions. Before you move on to the next question, because you are not going to be able to go back, reread the questions, make sure you answered what was asked. This thing is not designed to be a speed thing. You'll have enough time to finish. Assuming you're not like You're not going to have enough time to like take forever, but as long as you know this stuff, you'll have enough time to take care of it. So it's not going to help you to answer it like twice as fast as you need to, and you'll probably make a mistake by misreading a question. So reread the question, make sure you answer what was asked. Again, particularly in this weird world of online proctorio test taking. I'm going to be so happy when we have never looked at proctorio again in our lives. So that's another thing. Reread questions, make sure you answer what was asked. What else? I will be here for clarification. People, it often happens. Somebody, the Zoom lines are still open. People ask for clarification on something. You should never miss a question because you didn't quite understand what I was asking. You should only miss a question because you don't know the answer. So if you're not quite sure what I'm asking, you should definitely ask for clarification.
Would it be better to send you a chat message than to like just ask it, or do you have a
Either way, it can be chat, it can be, you can ask it as, unmute yourself. If every once in a while, if someone's completely off there, like someone might send an emergency like email through their phone or something. There is no notes or anything allowed. The system does check. It has you like do the little like, it was really absurd last time. Actually, last semester, I literally had, once somebody realized that there are these like thing where you have to like scan around your room, and I was like, don't just scan the ceiling. It was like, this person was, it was like, all right, this looks really freaking bad. It was like, so yeah, please, study well, but it doesn't help you. It doesn't help you to cheat, because it's just, everything you're learning now is really fundamental to the stuff you're going to be doing later on, and being, there's probably going to be a lot of nursing jobs now. I just read the Chronicle today just saying there's all these nurses quitting because they're totally burned out through COVID. So it's probably good for all of you in terms of like, once the pandemic is done and like everything settles down and all the current nurses have found other careers, there'll be lots of jobs available. No, I don't know, I'm being a little, I have to say, the people I know out in that world right now, it just sounds horrible and super stressful. And hopefully another few months, and more people are going to get their vaccines, and things are going to start feeling less freaky.
You were on semester going back to in-person yet?
So, I mean, at this point, we don't know. It's going to depend on a lot of things. I signed up. I had to make a choice. I said it's hopefully going to be hybrid, but that's going to depend on what actually is happening in the real world. So we're still not sure. I can say for sure that the next fall is not going to be normal for the most part, but how much we're going to try to bring back more in person versus not, it's not clear yet. And then reflexes, maybe we'll do those in breakout rooms on Tuesday after the exam. So don't worry about the reflex lab yet. Let's just do the reflex lab on Tuesday. You should just like focus right now on kind of getting ready for your exam, and I would say probably maybe starting the analysis of the enzyme stuff while it's fresh in your mind. But other than that, if you have questions about any of the material before Tuesday, email me. I'll do my best to answer any questions you have. And otherwise, have a good weekend.
So
So
we're
ju-
not doing the pre-lab for the next lab until like we're doing it together on Tuesday.
Yeah, exactly. I think that that makes sense because it's a pretty simple one anyway, and I think it makes more sense right now not to go off on some tangent and just stay focused on keep our eyes on the prize.
Thank you.
All right. Cool. All right. I will see you all on Tuesday.
Have a
Thank
good
you.
weekend.
Have a good
Thank you.
weekend. All right.
Thank you.
All right.